Thank you for joining us at the cutting edge of cardiovascular sciences. I'm your host, John Cooke. I'm chair of the Department of Cardiovascular Sciences here at Houston Methodist Hospital. And today, we're talking about engineering angiogenesis. Joining me today are my guests, Professor Ngan Huang from Stanford University and Professor Paolo Madeddu from Bristol. Let me just introduce them briefly. Ngan Huang is an assistant professor in the Department of Cardiothoracic Surgery at Stanford University. Her laboratory is at the Palo Alto VA Hospital. She received her BS in chemical engineering at MIT, and then from there went to get her PhD in bioengineering at UC Berkeley and UCSF, and then did a postdoc at Stanford University. She's going to be talking about her lab's work in engineering cardiovascular tissues. Dr. Paolo Madeddu is an internationally recognized expert in angiogenesis and vascular regeneration and bioengineering. He's chair of the Division of Experimental Cardiovascular Medicine at Bristol Heart Institute at the University of Bristol in England. So let's just ask the question, what is angiogenesis? I have just a few things to say about that before having our guests talk about their perspective. If I could go to the next slide here. There we go. So angiogenesis is an expansion of the microvasculature. Sprouting from existing capillaries is this new vasculature that is in response to poor blood flow, ischemia, hypoxia. You'll hear from Dr. Madeddu that there are many factors involved in this response, HIF1 alpha being principal, transcription factor that turns on a number of genes, including VEGF, vascular endothelial growth factor, which is one of the major angiogenic cytokines that plays a role in this process. VEGF induces proliferation and migration of endothelial cells, and that ultimately increases capillary density to enhance the microvasculature, improve microvascular delivery of oxygen and nutrients. So we'll be spending some time on angiogenesis today. There are some other processes involved in vascular regeneration. Arteriogenesis, the second panel there, is the positive remodeling of collateral channels from an occluded artery to restore blood flow. Adult vasculogenesis is the contribution of circulating blood cells to angiogenesis. Endothelial progenitor cells, a bit of a misnomer because they're largely hematopoietic in nature, but they do secrete angiogenic cytokines that contribute to the growth of the microvasculature. Perfusion can be restored by improving vasodilation, of course, prostacyclin and nitric oxide being primary endogenous vasodilatory compounds. Plaque regression can occur in conduit vessels where there's an occlusion or a narrowing, and that regression of plaque can improve blood flow as well. Somewhat more controversial is the contribution of therapeutic transdifferentiation. We won't be talking about that today, but it's an interesting new front in angiogenesis, and that is the transdifferentiation of fibroblasts into endothelial cells or something that behaves like endothelial cells to contribute to angiogenesis. Well, with that introduction, let me go to Dr. Ngan Huang to tell us a little bit about her work. Welcome, Ngan.
Hi, Dr. Cooke. Nice to meet you. Well, let me share my screen and so that we can talk a little bit about some of the work that our lab is doing in this area. Great. Great, and hopefully you can hear me and see my screen well.
Can hear you fine.
Great. Well, thank you for that background information. So as you may know, our lab is the lab that studies endothelial cells. And how we can utilize endothelial cells for various kinds of tissue revascularization, as well as treatment approaches for cardiovascular diseases. So first, by way of introduction, what are the endothelial cells? Many of you guys already know that the endothelial cells are the cells that lie in the inner lumen of all blood vessels. And they really are important cells to maintain the vascular homeostasis. In particular, endothelial cells are the cells that produce many factors that interact with circulating blood elements. And when those endothelial cells become dysfunctional, it can lead to a variety of diseases, including one that our lab studies heavily on, which is peripheral artery disease, or PAD. And PAD, many of you may know, is a disease that affects over 8 million patients in the US, particularly those over the age of 65. And although biological approaches have been tried to restore blood perfusion to these regions of tissue ischemia, the results have not been very successful. Cell-based approaches, such as the use of adult stem cell therapy, has only led to moderate benefit in the enhancement of revascularization. And so we were among the first labs to study an alternate cell source for treating peripheral artery disease, and this was using endothelial cells that are derived from these human-induced pluripotent stem cells, so we call it iPSC-derived endothelial cells, with the idea that maybe these could serve as an autologous source of therapeutic endothelial cells that could be injected into sites of ischemia to induce angiogenesis. And so we first tested the efficacy of these cells in a mouse model of PAD, which we call the high-limb ischemia model, because it's a model where we essentially ligate and excise the femoral artery, which is the main artery that supplies blood flow down the leg. And by excising this femoral artery, it leads to an instant reduction in blood flow by more than 80%. And so this was a model for us to be able to study how different kinds of cell therapies may be able to improve revascularization and reperfusion in the ischemic limb. For purposes of labeling the cells so that we can track them using bioluminescence imaging as well as fluorescence imaging, we use a viral construct, and we're able to generate endothelial cells that express GFP as well as luciferase. And in this model, we injected them into the mice with high-limb ischemia. And what we found surprisingly was that the cells pretty much went away really rapidly over the course of a few days after transplantation. And so in the end, in order to have a therapeutic effect, we found that we needed to inject cells twice, 500,000 cells at the time of induction of ischemia, and again, another dose of 500,000 cells after 7 days. And of course, this did have some benefit in terms of improving their perfusion. And this was shown by a technique called laser Doppler spectroscopy. But we really were concerned about the poor cell survival post-implantation. And so that's what led our lab to be interested in trying to engineer angiogenesis by utilizing cues from the extracellular matrix. And one thing I had been really fascinated about for many years is the morphology of endothelial cells within healthy blood vessel segments. And so an image like this is one from Peter Davies' group that's really well established, showing that the endothelial cells in really healthy laminar flow environments have an elongated morphology that where the elongation is oriented along the direction of flow. And so we thought that maybe this kind of cell patterning is really important for maintaining their function. And the reason is that in the regions that are atheroprone, which tend to be in the regions of bends, branches, and bifurcations, those endothelial cells lose their morphology and have sort of this randomly oriented appearance. And so we thought that maybe spatial patterning of the extracellular matrix can be a way to pattern the cells, and whether or not patterning the cells to mimic this sort of healthy, elongated, parallel aligned cellular morphology, whether or not it could have some benefit for enhancing cell function as well as their survival. And so we worked with an industry collaborator to develop spatially patterned scaffolds that have nanoscale spatial grooves. And so you can see here from these atomic force microscopy images that they are collagen, so they're made of collagen, but the fibrils are on the nanoscale of about 30 to 50 nanometers in diameter. And they're prepared by a facile technique that takes advantage of the fact that collagen is as a monomer is in a highly acidic environment. But if we extrude the collagen, in this case as a film, using shear, that induces fibrillogenesis, especially if they are soaked into a pH neutral environment. That neutral environment causes the fibrillogenesis to be induced, and that direction of shear induces the fibrils to form in a highly organized way, leading to the formation of these films that have parallel aligned fibrils, in this case with even a little bit of waviness. And these films then can self-assemble and form these long and thin threads, as shown here, that are very similar to the dimensions of suture. And when cells, in this case endothelial cells, are grown on the exterior of these scaffolds, those endothelial cells miraculously have a very spatially patterned appearance. This is shown here by F-actin staining of their actin cytoskeleton, as well as by using scanning electron microscopy, where we can see these endothelial cells that are highly elongated and parallel aligned, very similar to how those healthy endothelial cells look in the previous image. And so we were able to develop a very reproducible method for getting endothelial cells to be aligned on the exterior of these very long and thin scaffolds. And so we wondered if we implanted cells, not just by injecting them in saline, but rather giving them an instructive biomaterial that provides these spatial patterning cues, could we, in that case, improve their survival or their angiogenic function? And so we tested this out in our same hind limb ischemia model. And our goal was to determine if these essentially what we call parallel aligned nanofibrillar scaffolds can guide endothelial cell assembly to bridge the area of tissue ischemia and be able to enhance and guide the formation of oriented microvasculature. And so in initial studies, we first looked at just using primary endothelial cells that were delivered on either our aligned scaffolds or on what we call a conventional randomly oriented collagen fibrillar scaffold. And as reference controls, we also had treatment groups that were just acellular or cells injected in PBS as a standard, as well as a PBS only kind of control group. And what we found is that the survival of the cells when grown on the aligned scaffolds were remarkably better compared to cells that were just purely injected in saline or salines on cells on a non-patterned scaffold. And although you can see by the quantification as shown here on the right, there is a drop in survival over time, but we were able to still maintain the visibility of cells over the course of 14 days. The other thing to also that will help us appreciate this data is that because the cells are growing only on the exterior of the scaffold, where the scaffolds are very long and thin with a diameter of no more than about 300 microns, there are no more than 10,000 cells on a 1 centimeter long piece of scaffold. So this is 10 to the 4th cells, whereas in our previous initial studies, we delivered 10 to the 6th cells. So this is 2 orders of magnitude less cells that we're delivering, yet we still see a signal after 14 days. So this was really showing us that the scaffolds themselves were able to enhance and maintain the survival of these cells presumably by providing an instructive biomaterial environment for the cells to survive in. And in part, because the cells were able to survive longer, we were able to show using laser Doppler spectroscopy that the perfusion recovery was significantly better when the cells were growing on the aligned scaffold as well. And this is quantitated by what we call the mean perfusion ratio, which is the perfusion in the ischemic limb divided by that of the control limb. So every animal has its own reference control. And so we found that the cells growing on the aligned scaffolds were able to significantly restore blood perfusion over the course of 14 days. Now, we repeated the same experiments using our iPSC-derived endothelial cells and also utilize a new imaging approach to be able to look at the microvasculature. And this is work that continues from our earlier studies in Nature Medicine, where we developed and tested alongside with Dr. Hanja Dai, our collaborator in the Department of Chemistry, intrinsically autofluorescent fluorophores. In this case, these are fluorophores composed of carbon nanotubes that intrinsically autofluoresce in the near infrared second window, so over 1400 nanometers of wavelength, where there's very little endogenous autofluorescence. And so by injecting these carbon nanotubes systemically, they serve essentially as vascular contrast dyes that allows us to look at the vasculature that is perfused. And in doing so, we're able to use these dyes as a way to visualize the microvasculature and found it using a quantitative a measurement of vascular density, that the iPSC-derived endothelial cells growing on the aligned scaffold was able to have also a significant improvement in vascular density compared to, again, cells that are just delivered in saline. And so we thought this was really exciting that these spatially patterned scaffolds were able to have an angiogenic effect as well as a pro-survival effect. And some of our ongoing work, which is not published yet, is really to try to understand some of the fundamental biology of what is at play. And so most recently, we started performing a bulk RNA sequencing, comparing these cells, these iPSC-derived endothelial cells on aligned scaffolds compared to that on randomly oriented collagen scaffolds, to try to understand what kind of gene expression profiles distinguish them. And long story short, there are over 1,400 genes that are differentially expressed. And among them, when we performed some gene set enrichment analysis, we were able to identify some of these pathways that are related to cell matrix interactions, including regulation of cell substrate adhesion, cytoskeletal organization, and regulation of focal adhesion assembly. So this is just some of the ongoing work that will help us to elucidate exactly what are the mechanisms by which these spatial patterning cues are influencing endothelial cell function as well as survival. And so this is an area of ongoing work in our lab. But when we started to create these biomaterials for delivering cells, we also thought about, well, maybe in some cases, it's not enough to just deliver cells on a scaffold and expect the cells to know what to do in terms of making their vasculature. Maybe we need a more comprehensive approach. And in thinking that in PAD, not only is there endothelial dysfunction, but in some cases there's also dysfunction of the muscle tissue. And so we start to think about how to create an entire engineered muscle with healthy muscle that is vascularized. And we had the idea of mimicking the natural physiological organization of skeletal muscle, which is generally speaking, myofibers in parallel, interdispersed with capillaries that are also in parallel. And so in thinking about our scaffolds that are very long and thin, we thought, okay, this can be approach for us to deliver essentially bundles of muscle fibers interspersed with endothelial cells as a potential way to treat PAD. And so to begin with, we first determined whether or not the spatial patterning is able to induce not only endothelial cell organization, but also skeletal muscle organization. In some of these early studies, we used as a proof of principle mouse C2C12 myoblast cell lines, but later validated it using primary human myoblasts as well. But in these studies, we co-cultured endothelial cells with skeletal muscle myoblasts on either aligned or randomly oriented scaffolds. And we also compared them to the case of just myoblasts alone in the absence of the co-culture. And remarkably, we find that the aligned scaffolds are able to also provide these spatial patterning cues that allow the myoblasts to become aligned along the direction of these fibrils, and therefore also be able to help them in a facile way form aligned multinucleated myotubes. And this is shown here in confocal images of myosin heavy chain as well as CD31, where the CD31 is labeling the endothelial cells and the myosin heavy chain is labeling the skeletal muscle myotubes. And the myotubes in particular were much longer on the aligned scaffolds compared to that of the randomly oriented ones. And actually co-culturing, having the myoblasts with the endothelial cells, are able to significantly increase myotube length even more. And what you can appreciate down here are some of our functional measurements by looking at skeletal muscle contractility in response to electrical stimulation at 1 Hz, where we're able to see that with the alignment of these myotubes, they're able to contract in a much more synchronous kind of manner. Well, we also want to know, do the endothelial cells also help the myoblasts in some kind of paracrine kind of interaction? And so what we did was that we looked at the proteomic analysis of endothelial cell secreted cytokines when they are co-cultured with myoblasts. either on the aligned scaffolds or on the randomly oriented scaffolds. And sure enough, we found a number of cytokines that are produced significantly in the presence of the aligned scaffolds compared to the randomly oriented scaffolds. And some of those cytokines included angiogenin, IGF binding protein 3, as well as VEGF, one of our favorites. So it was really showing that on the aligned scaffolds, the endothelial cells were better at producing pro-angiogenic and sometimes pro-myogenic factors. And I think this kind of interaction is important for ultimately for muscle regeneration as well as revascularization. But just as a proof of principle, so this is showing here some of our in vivo testing of what we call our endothelialized muscle bundles. And so in the same way we made those long and thin scaffolds that are seeded with myoblasts and endothelial cells, well, we bundled 8 of them together to sort of mimic how a muscle bundle looks, and implanted them into a mouse model where it's implanted into the tibialis anterior muscle. Because this muscle is a bit bulky, we did have to remove some of the existing muscle. So we did ablate some of the muscle in a normal mouse and then implant our engineered muscle in its place, and looked at the effect of having the co-culture of endothelial cells as well as myoblasts on either the aligned or randomly oriented scaffolds. And one of the things that we found really intriguing was, at least on the muscle side, that the aligned scaffolds were really able to improve the survival as well as proliferation of these cells over time. And these are experiments where we specifically bioluminescently tagged the myoblast population, showing that on the aligned scaffolds in co-culture, those myoblasts had the highest degree of cell survival and proliferation over the course of 21 days. But we were also really interested in what happened to the endothelial cells and how does the organization of the vasculature looked. And so we performed histological analysis of that. So first we looked at cross-sectional, so transverse slices of our engineered tissue implants. And because the muscle, the myoblasts were, in this case, GFP labeled, we can track these transplant-derived muscle myofibers. And we know that they're muscle myofibers because they co-express myosin heavy chain and have centrally located nuclei. So these are all hallmarks of newly formed muscle fibers that came from the transplanted cells, shown here in green. And sure enough, we do see a lot more transplant-derived newly formed myofibers when they are cultured on the aligned scaffolds. But interestingly, we also were very happy to find that the spatial patterning of the aligned scaffolds were also able to help form the organization of the vasculature as well as the myofibers. And this is shown here in the confocal images, showing that near the aligned scaffolds, those vasculatures there are all patterned in a parallel aligned way along the direction of the fibrils on the aligned scaffolds compared to those on the randomly oriented scaffolds. And in this sort of 3D reconstruction, one can also appreciate here these GFP positive transplant-derived myofibers are essentially in parallel and interspersed with also aligned capillaries. And so this very much mimicked one of the earlier pictures I showed, which was the physiological organization of skeletal muscle myofibers interspersed with parallel line capillaries. And so this was beginning to look more like that physiological organization. And so I think with that, this is some of our work that we'd like to talk about, related to using spatial patterning as a way to provide ECM cues to enhance endothelial cell survival and angiogenic function, as well as taking it to the next step of trying to actually use spatial patterning as a way to create spatially oriented skeletal muscle interspersed with spatially patterned microvasculature.
Nan, that was great. A striking demonstration of the importance of the extracellular matrix patterning. We're going to take just a couple of questions. I just want people watching to know that you can call in. You can join us by text. You can text your question to DEBAKEY 37607. and then text in your message. Or you can go join by web, go to polev.com and enter DEBAKEY. Then you can ask us some questions. Nan, I have one for you. You showed us some beautiful immunohistochemistry. What about perfusion, though? Do these vessels that are nano-aligned, are they carrying blood flow? Are they improving perfusion?
Sure. And in the earlier study that I showed that was involving peripheral artery disease, so in the high limb ischemia model, when we injected the fluorophore that is emitting in the near infrared, because it's injected systemically, it is systematically only labeling the perfused vessels. In the latter study, where we transplanted the engineered muscle into the tibialis anterior muscle, because that one is not an ischemia model per se, this was more of a demonstration by basically ablating the muscle tissue to make space for implanting this engineered tissue. This particular study, we didn't quantify blood perfusion per se, but perfusion recovery, because it's not an ischemia model. But we did look at the perfusion of the microvasculature through the isolectin staining. So right before sacrificing the animals, we injected the animals with isolectin systemically. So the isolectin binds to endothelium of perfused vessels. So certainly we've quantified the number of perfused vessels in our engineered tissue, showing that there was a significantly higher number of perfused vascular density when the transplants consisted of myoblasts and endothelial cells in co-culture on an aligned scaffold. But we did not look at, for example, laser Doppler, data, only because that model is not an ischemia model.
Thanks.
Ngan, this is Paolo speaking. A question for you. You started to show that the cells incorporated in the scaffold are somehow protected from clearance. Do you think that they are physically protected from macrophage? And also, I would be quite interested to know what is the macrophage and recruiter cell behavior. So if the scaffold itself can produce or stimulate angiogenesis through recruitment of angiogenic cells.
Sure. So in our control groups, where we just look at acellular scaffolds, we find that there are very few newly formed vessels. In all cases, the scaffolds are fairly biocompatible, so there's not any acute inflammation, but there are macrophages that are associated with the hind limb ischemia. So, of course, in response to injury or disease, there are some macrophages, but nothing extraordinary compared to that would suggest fibrosis or that the biomaterials themselves are not biocompatible. But in terms of maybe the other question that you may be getting at, which is, do these spatially patterned scaffolds actually regulate the macrophages themselves? So that's a very good question, because in all of our cases, we primarily focus at the endpoint. So by then, the macrophage population is probably not significant at that time. But at least in the literature, there are data to suggest that they can also interact with the macrophages, and that could also be another mechanism as well. But we just have not looked at those earlier time points.
Thanks, Nan. I want to give Paolo enough time to talk. He has some interesting things to tell us about genes, gene therapy, cell therapy for ischemia. And it's a nice follow-on from your talk, Nan, about the importance of nanopatterning the matrix. Paolo, let's go to your talk.
Yeah, I don't know if you can see my screen.
You can see it fine.
Okay, so actually I obviously share the enthusiasm of the previous speaker about the tissue engineering. I want to make a step back to the different components that led to the idea of tissue engineering. So tissue engineering is the very last step in the different approaches that were attempted in angiogenesis therapy, which is encompass a spectrum of strategies. The first is certainly gene therapy, which is historically was used to stimulate therapeutic angiogenesis. More recently, cell therapy, And a version of gene therapy is a direct delivery of angiogenic protein or eventually small molecules, and also eventually microRNA, which I consider small molecules. And the target is not only the ischemic limb, as shown by previous speaker, but also improvement of the circulation, the coronary circulation is the infarcted heart, and improve also the perfusion of failing heart. So the application is a heart multiple, acute condition requiring improvement in perfusion, but also chronic condition in which improve the perfusion can also have a benefit, exert a benefit on contractility of the heart. So just making just playing on my personal experience. I'm coming from a different field. So in around 2000, I changed my interest clinical arterial hypertension to angiogenesis, and it was just a shift due to a collaboration with a colleague now in the US, Maurizio Capogrossi. He was building an institute in Rome, and I started collaborating with him on the possibilities that kallikrein, which is an enzyme which cleave bioactive product called kinase, could eventually inhibit intima hyperplasia, so a complication after revascularization. And since Maurizio was quite interested in angiogenesis, I started thinking that because kinases are peptides that activate the nitric oxide pathway, this enzyme, kallikrein, which generates kinase, can be eventually angiogenic. And this actually led to a serious study confirming our initial hypothesis. So we injected the kallikrein as a gene using an adenoviral vector, and kallikrein was able to generate a kinase in excess and stimulate both the B1 and B2 receptor. And this activated intracellular signaling centered on AKT and nitric oxide, but also stimulation of other pro-angiogenic factors, including VEGF. You can see here an image of the collateral formation in a limb ischemia model as compared with control. And as histological evidence that kallikrein was able to not only improve the number of capillaries, but result in a much mature vascularization made by endothelial cell, vascular muscle cell, and surrounding pericyte. The evolution of this project led also to clinical treatment. In the last 5 years, the kallikrein formulation of kallikrein protein was used in China to treat patients with stroke, and is now approved in China as a clinical product. It's also trialed in the US for the same application, and I believe that this could be also, there is possibility for kallikrein therapy in other conditions such as myocardial infarction and limb ischemia. The other factor that we studied was NGF. It's a neurotrophin that is implicated in neuronal survival and repair. And the group led by Costanza Manueli, now professor at Imperial College in London, together with Andrea Caporali and Meloni, who are now relocated in Edinburgh, was able to demonstrate that neurotrophin, in particular NGF, stimulate also a pathway centered on VEGF and AKT and enhances the activity of endothelial nitric oxide, leading to endothelial cell survival, proliferation, and migration, and finally angiogenesis. The nice part of this work was also that NGF could act on the bone marrow, activating the release of pro-angiogenic bone marrow cell, and this could lead to potentiation of local angiogenesis, so recruitment of this cell into the ischemic tissue, together with also repair on ischemic nerves. So just to give you an idea of the clinical translation of this research, NGF is now used in clinical trials to treat cutaneous pressure ulcer, particularly diabetes, corneal ulcer, glaucoma, and retinal maculopathy. But in the following years, we also try to shift the concept because most of the therapeutic angiogenesis was trying to correct some kind of pathogenetic mechanism. So in other words, in disease condition, you have a lack of some angiogenic factor. As the initial idea of this field was try to replenish ischemic tissue with a selecting factor. So in other words, is a pathogenic approach. However, we thought that we can eventually reverse the concept. Instead of correcting the disease, we wanted to eventually test the possibility of introducing in the ischemic tissue the successful effect of a gene that is eventually working very well in centenarian. So, there is a lot of studies about the genetic component of health span and prolonged lifespan. And our group discovered, in collaboration with Professor Annibale Puca, the one polymorphic variant associated with prolonged exceptional longevity can eventually activate the angiogenic project. We tested this in limb ischemia, myocardial infarction, and more recently in diabetic cardiomyopathy. In all the cases, this approach was extremely valid and induced not only an improvement in angiogenesis, arteriogenesis, but also improvement in contractility. The difference in terms of molecular pathway is that this polymorphic variant activate several mechanisms, including a better behavior of nuclear protein. It also activates the clearance of exhausted protein and also inhibit oxidative stress as a level of mitochondria. We also found that this polymorphic variant can create a beneficial polarization of macrophage M1 and M2. And more and more is coming out of this particular research. So we believe that this particular gene therapy could be beneficial in defect angiogenesis related to aging. Another great interest in our lab is focusing on pericyte. So pericyte are mural cells that surround the capillaries and arterioles and are recruited during sprouting angiogenesis to stabilize the nascent vascularization. We approached this particular strategy in different way. Initially, simply injecting pericytes that were isolated from different organs. The first isolation from small leftover of cardiac surgery by Dr. Campagnolo, and then apply pericyte cell therapy, particularly with work done by Dr. Catare in model of myocardial ischemia, initially in mice and later on in large animal model. But more recently, there is a particular strain of research that is conducted now independently by Dr. Avolio, who is trying to use small molecule to create a kind of in vivo transdifferentiation of a pericyte into contractile vascular muscle cell. In this way, also promote arteriogenesis. This moment, the originality of Elisa's work consists in the fact that finally he found a specific marker to identify pericyte and distinguish them from contractile vascular muscle cell. And she also using small molecules to stimulate the pericyte into this conversion. Interestingly, she also found that the process is reversible. So we have a kind of antidote to eventually let the phenotype come back to the original phenotype. So The next aspect that I wanted to cover in my presentation is not overlapping with the previous one, because our applications are twofold. We also generate vascular scaffold that can be applied in two conditions. The first condition is congenital heart disease. The second condition is grafting a small arteriole less than 5 millimeters in diameter. So we use different kind of cell to this approach. Obviously, our favorite cell type are the pericyte, but we are also interested about other mesenchymal stem cells that we can collect from the umbilical cord, for instance. And the cells are seeded in artificial scaffold, but also in xenogenic scaffold before implantation in animal model. Together, this form what is called advanced therapy medical product that I will call later ATMP, which is subjected to regulatory approval. So we needed to create a system that are not only treated and tested in animal, but later on also viable for clinical application. So together with the neonatal cardiac surgeon, Professor Caputo, we are able to identify the better population, the best population according to the diagnosis of congenital heart disease. The diagnosis is done prenatally. We generally prefer use IPS derived from tissue of the fetus. Otherwise, if the diagnosis is done postnatally, we eventually rely on materials such as the cord, or eventually also leftover from cardiac surgery. The autologous cells are then seeded on xenogenic scaffolds that the cardiac surgeon is using for correction of the defect. And then the advantage of this procedure is that we believe that the scaffold can grow together with the growth of the baby. So this actually is the first step of the isolation of the tissue from saphenous vein, of the cell from saphenous vein, myocardial sample, umbilical cord. We use immunomagnetic separation and then isolation and selection through selective culture system. The next step is seeding the cell on the matrix, which it could be a porous scaffold or xenogenic graft, and then a low maturation in a bioreactor. This actually is a result of an experiment done in using a xenogenic scaffold that was decorated with pericyte from pigs. And this actually 6 months after implantation. You can see that the seeded scaffold have a thicker endothelial layer, and also the position of the endothelial cell in the endothelial layer is oriented according to the flow, as previously shown by the other speaker. The other important aspect that we saw after a recovery of the scaffold was an improvement in angiogenesis in the tunica media. This is absolutely important because we need a tissue that is well perfused to mature and be consistent, viable after months from implantation in the tissue. The other interesting data that I want to show you now is actually an unpublished result of showing that the endothelium is not only better presented after implantation of a scaffold seeded with pericyte, but also express a larger amount of nitric oxide. And this is particularly important in the case of replacement of the pulmonary artery, because this is an area that is low pressure. So we need low resistance, and we need a larger production of nitric oxide. So the presence of pericyte in the scaffold enables the scaffold itself to release a larger amount of nitric oxide and prevent eventually the development of arterial pulmonary hypertension, which is a major complication in a patient with congenital heart defect. And finally, I want to show you some preliminary data that somehow are related to the previous presentation. As shown before, it's essential to have endothelial cell inside the scaffold, but we're trying also to generate a more complex structure that is made by synthetic material and gelatin. So it's a kind of hybrid, and in which the endothelial cell are decorating the intima, and then mural cell, so those muscle cell and pericyte are then printed in the external area. This material is then better mature in a bioreactor, and our next step will be to implant in a animal model, large animal model of carotid occlusion. So this work is done mostly by Caraba, Fagnano, and Presutti, who is also studying a mathematical model to improve the hemodynamics of the scaffold. So I believe that I wanted to conclude here, showing that we have actually several possibilities to work on tissue engineering, but in the meantime, it's also very important that we refine the other aspect of therapeutic angiogenesis, as I said before, particularly which kind of angiogenic factor we want to use, which kind of cell we want to use it for decorating this vascular graft. And I believe that the synergy between different disciplines will really lead to a final result that is clinical translatable. Thank you.
Thank you, Paolo. That was a tremendous tour de force. You took us from angiogenesis all the way to conduits, bioengineering, cardiovascular tissues. That was spectacular. We're going to take some questions in a minute, but I just want to remind people, if you want to join by text DEBAKEY to 37607. Alternatively, you can join us by web. Go to polev.com and enter DEBAKEY. Paolo, you're a physician as well as a scientist. You have an internal medicine degree. You are a cardiologist. Where are we now? If you were to survey the literature and survey what's going on today in cardiovascular bioengineering, how close are we to getting a truly cardiovascular bioengineered tissue into the clinic?
This is a very important question. To be honest, I worked for a long time believing that this perded stem cell could eventually be the solution. But I was disappointed by the fact that they last a very short time. So I don't think that injection of cell will lead really to tangible result. On the other side, I believe that there is a big potential for the generation of small vessel by the methodologies that we show today. And the other possible scenario that will, clinical scenario, will benefit from tissue engineering, in particular congenital disease. And the reason for that is because a combination of really addressing an important aspect, so allowing the graft to grow with the baby, but also deal with the ability of the youngest individual to have a strong regenerative potential. So this together will make a strong difference. Now, related to what is the next challenge with the tissue engineering, I believe that we need really to understand what are the best cells. And one major question is, should we go with autologous or allogenic? The limitation of autologous is obviously we can treat a single patient, but it will be difficult to propose this to the NHS as a treatment for all the patients that need this kind of intervention. So this actually is a provocative question for Ngan. What is the next step for you? What do you think that not only will allow you to win the next grant, but also, to make a translational step in your work.
Sure. Dr. Madeddu, you presented, really great, slides on how the different technologies are also a very important element of being able to have these different scientific advancements. And I think right now we're at the cusp of being able to utilize advanced technologies, with 3D bioprinting, as well as other advanced chemistries, to be able to allow cells to see much more complex cues, whether or not they're spatial or geometric cues, or also like gradients of paracrine factors or genes. And so I think the next frontier is really being able to synthesize all of the different, specific assays that we've developed and integrate them in a much more complex engineered tissue. So for example, utilizing some of the newer 3D bioprinting strategies to create much more complicated structures, as well as intercellular interactions and different populations, but then potentially also taking advantage of different chemistries to be able to have different, gradients in soluble factors, and taking advantage of some of the gene therapy approaches. Maybe it's CRISPR based, or maybe it's just a straight, gene delivery approach. But I think that, if we can create a more complex system that better mimics the true complexity of the native system, then I think we'll be a little bit closer to trying to realize, our goal of ultimately being able to create a completely bioengineered artificial myocardium.
Mm-hmm. And the forgotten circulation, the lymphatic circulation, also is in need of regenerative therapies. Patients with lymphedema, for example, women who are post-mastectomy and have lymphedema, need a solution. And it seems like the threads that you're using may lend themselves to lymphatic reconstruction. Can you talk about that?
Sure. This is some of the work that we have applied using a porcine model of lymphedema, where these long strands were allowed to connect between lymphatic collectors in an area that is obstructed. And so to mimic that area of lymphatic obstruction, we performed lymph node transfer removals, as well as performed irradiation. So very similar to sort of a cancer kind of treatment environment to induce lymphedema. But then once the lymphedema was developed, to use our spatially patterned scaffolds to surgically connect two ends of our scaffold to lymphatic collectors. And we were, really, happily surprised to see, the formation of new lymphatic vessels and collectors using this kind of scaffold-based approach. And these scaffolds also were shown to also have the benefit of releasing pro-lymph angiogenic factors like VEGF-C, was also able to in a localized manner, recruit the formation of lymphatics within the vicinity of the scaffold. So we think that these spatially patterned scaffolds are able to induce vessel formation of both blood vessels as well as lymphatic vessels.
And I guess we'll know soon because there's a clinical trial ongoing with these threads. Wonderful, that's wonderful. Paolo, you had another comment?
Yeah, I have a question also for Ngan, and I consideration. So I was wondering and ask myself, and then now ask you, what do you think though that this graft containing human cell can be also used for a screening on new angiogenic substances and create this kind of modeling that is probably superior to the animal model because we can use human cell to decorate the graft.
Yes, I believe that's definitely a great way to be more efficient than using animal studies. I think there is value to both, but I completely agree that using a more of a systematic in vitro approach of vascular grafts could be a great way to screen for initial candidates and maybe sort of validating them with animal models down the line. So I think the screening approach is a great first step.
It's a very complex, isn't it? Bioengineering an organ. One of the factors that you mentioned, Ngan, just briefly, and I wonder if you could elaborate a little bit more on this, is the issue of the matrix stiffness. how does that affect cell replication, cell phenotype?
Sure. So cell stiffness is actually a very interesting area for us because we want to make scaffolds that can provide really the right signals. And we know that stiffness makes a very important difference. And so in our studies with our collagen-based scaffolds, we're able to tweak the stiffness of the biomaterials by using different kinds of cross-linking chemistry to vary the stiffness. And so at least within the range that we've tested, which comes from a small range, the stiffness does have some effect on cell biology, but I think that our range was not a very wide range, so we really only looked at the range of maybe one order of difference. But in some of our more recent work, where we were just looking at the effect of substrate stiffness in general on just endothelial cells cultured on hydrogels of varying stiffness, we see that the stiffness of the hydrogels are really able to have a big effect on a slightly other area, which is equally important, which is what we call endothelial to mesenchymal transition. And finding that when we grow endothelial cells on our convention polystyrene dishes, those kinds of very stiff, pathologically stiff environments are really bad at maintaining endothelial cell phenotype. And that's probably one of the reasons why when we typically grow primary endothelial cells, we only grow them for a finite number of passages. And it's not only because they become senescent, but also we feel that some of those cells are actually taking on more of a mesenchymal kind of phenotype. But when we grow them in much softer, physiologically kind of relevant stiffness gels, which are on the order of about 4 to 10 kilopascals, those endothelial cells, even when induced with cytokines and factors that normally trigger endo-MT, when cultured on those soft hydrogels, endothelial cells are less prone to undergoing endo-MT. So there we are really beginning to see a major difference in endo-MT.
That is very interesting.
I also agree. I agree that apart from endothelial cell, obviously mural cell and pericyte are also very sensitive to mechanical strain, and they can contribute to the creation of the extracellular matrix. So, for instance, we screened the secretome of human pericyte, the cardiac pericyte, and we found that they secrete a large amount of extracellular proteins such as SPARC1. And interesting enough, SPARC1 is also an angiogenic factor. So pericyte can modulate themselves the extracellular environment and contribute to production of extracellular matrix protein. But also through SPARC1, they can induce angiogenesis. So it's a kind of reverberating phenomenon where the matrix influences mural cell and vice versa, the pericyte mural cell can contribute to the formation of the extracellular matrix. So it's a really fascinating aspect.
Guys, we have one question that I'd like to ask you from the audience before we call it quits. One of the audience members are asking, where could I find a trial to get involved with, a bioengineering trial? I guess it depends upon what organ you're bioengineering and where you live, but I suppose, my answer to that would be clinicaltrials.gov might be a place to go to. You can search for a particular disease and look to see what's available there. How about in your neck of the woods in Bristol, what would you advise people to do?
Yeah, actually, in this moment, I can tell you what we are doing in Bristol. We are applying for a clinical trial in case of patient with pulmonary atresia. So we obtained already some approval from a regulatory agency, and we are just asking the British Foundation to sponsor this project. If funded, we will be able to do a safety trial in babies with tetralogy of Fallot using a xenogenic material that is decorated with a cord mesenchymal stem cell. So this will be a trial in babies, and I believe that there will be really high chance to be successful.
Thank you, Paolo. Well, I want to thank you both for joining me. Paolo, you're a hero. It's late there in Bristol, and I appreciate you staying up late to talk to us. Ngan, so nice to see you again. And both of you did a wonderful job. This was very exciting, I think, for me and for everyone watching. So thank you again for joining us, and hope to see you again soon.
Bye.
Thank you. Take care.
Okay.